and welcome to another virtual episode of NASA Science Live. I'm your host, Lauren Ward, and today we have some very exciting news for you. Scientists have created thick, vascularized human organ tissue in a lab. These are the first revolutionary steps to 3D printing functional human organs. So you may be thinking, what does NASA have to do with this, and why is it such a big deal? Stay tuned, because today you're going to find out all about this and more.
It will have an enormous impact on humankind.
It can really change the face of medicine as we know it.
The vascular tissue challenge aims to tackle one of the most fundamental problems in the field of tissue engineering.
The vascular tissue challenge is the Centennial Challenges prize that's given to the first 3 teams that are able to create
Vascularized tissue, that is to say, tissue with its own blood vessels,
so that we can actually grow complex tissues that are required for whole organ development.
People can make veins, people can make arteries, but no one can put all of it together.
The huge challenge is to ensure that this tissue functions.
we need to be able to get nutrients in and get waste products out.
There needs to be a blood supply.
NASA Centennial Challenge is a really unique way of tackling certain problems.
It has the capacity to revolutionize organ transplantation, tissue regeneration. We can make a difference both on Earth and in space.
Teams across the country have been competing for years to be the first to engineer functional human tissue. And today we're going to announce the official winner of NASA's Vascular Tissue Challenge. Here to talk more about the challenge is Monsi Rahman, the program manager of NASA's Centennial Challenges. Monsi, thank you so much for joining us today.
Thank you, Lauren, for having me today. I am so excited to talk about the winner and what we're doing.
Yeah, there is a lot to be excited about. Okay, basics. What is the Vascular Tissue Challenge all about?
The Vascular Tissue Challenge is about incentivizing the public to help us develop thick, functional tissue from major human organs that can one day perhaps help us achieve long duration missions, keep our astronauts healthy, and also have amazing Earth applications.
Amazing. That sounds great to me. Why is NASA interested in this sort of revolutionary technology? You touched on it a little bit, but let's go a little bit deeper.
So NASA is super interested in this for very many different reasons. So the first one, short term, would be the possibility of help incentivize the commercialization of the International Space Station by allowing companies to perhaps create tissues for humans on Earth that need a little bit of help with their organs that might be not functioning 100%. But in the long run, we are looking at these kinds of technologies to help us understand the effects of radiation on the human body without using human bodies, using tissues, and then long term after that, possibly to help us maintain the health of our crew when we go to Mars and beyond.
Wow, that's amazing. So I understand this is just one of many prize competitions at NASA. Why challenges?
Oh my goodness. Challenges are amazing. It is a way to involve the public in the NASA mission and allows people that perhaps had never thought about competing or about being part of the NASA groups to be part of our group. We have had an incredible success with people that have never ever even dreamed about contributing to the NASA mission. And challenges are a way of doing that. It's a way for us to tell you what our problems are, our issues are, our technology gaps, and for the public to come back and tell us, the amazing things they're doing and how their technologies can help us.
Wow, that is incredible stuff. Thank you so much for joining us today, Monsi.
You're welcome. Thank you for having us, and we're super excited.
So solving the Vascular Tissue Challenge will have a huge impact on NASA's long-term goals, but it can also have an impact on those of us here on Earth.
The Vascular Tissue Challenge wants to make a piece of tissue.
Vascularized tissue is a composite type of tissue. It has more than one sort of cell in it.
There need to be blood vessels throughout this tissue that are integrated fully with the organ tissue.
We've seen some great successes in the development of tissue engineering products.
We are making some progress with regard to vascularization. We're beginning to understand the signals that are necessary to switch on cells to become blood vessels.
If we could create vascularized tissue, we could create whole organs. In order to successfully grow whole organs, it'll be essential that we further develop these technologies.
Putting all those together is really the holy grail. That's what we're looking to do.
And the vascular tissue challenge is an essential step that will help to address that issue.
This is definitely possible.
To help us understand the significant impacts this challenge will have on the scientific and medical communities, we are joined by Dr. Arun Sharma, Vascular Tissue Challenge Judge and a Research Fellow at Cedars-Sinai Medical Center. Thank you so much for joining us today.
Thank you for having me.
Okay, one thing I think we should clear up here is, can you explain exactly what vascularized tissue is?
Sure, I'd love to. Vascularized tissue, in the simplest term, is tissue that has blood vessels. And the reality is all the tissues in our body are comprised of many, many blood vessels. So if we want to better understand how the human body is functioning, we want to develop vascularized tissues outside of the body that also contain engineered blood vessels.
Wow. Okay, so it's a crucial component to basically, it's one of the building blocks, it sounds like, to doing more research on this topic. But why is it important that we engineer vascularized tissue? What makes it such a difficult challenge to solve?
Certainly. Well, vascularized tissue enables tissue in general to get bigger and better. Blood vessels supply the nutrients and oxygen that all tissues in the body need to grow and function appropriately. And so if we want to better understand how the human body is actually functioning, we wanted to engineer true vascularized tissues outside of the body. And these tissues ideally should have the many, many blood vessels that are found in all tissues in the human body. It's not an easy thing to do because, as a lot of us know, the human body has hundreds of thousands of different capillaries, blood vessels, arteries, veins, all different types of vessels that are important for supplying the nutrients to the body. And a lot of times, nature is the best engineer, and we're trying to replicate what nature can do by introducing as many of these blood vessels as we can in these tissues outside of the body.
Wow. Okay, so looking forward, how will this change medicine as we know it?
Absolutely. I think this is going to change medicine in two ways. One is that these vascularized tissues will allow us to better understand how the variety of different tissues in the human body actually function. As I mentioned, all tissues in the body have blood vessels. So it doesn't matter what disease you're interested in studying, whether it's cardiovascular disease, liver disease, brain disease, all diseases of the body are intersected with the circulatory system and the blood vessel network. So if we can engineer better vascularized tissue, we can study a variety of different diseases in a dish, so to say. And the second reason why this is really important is because down the road, we want to potentially use some of this vascularized tissue for therapeutic purposes. Just to give a simple example, after folks have a heart attack or a myocardial infarction, as we call it, a lot of the cells in your heart are lost. And ideally, you want to be able to replace those lost cells with an adequate cell type that's going to replace the function of those dead cells. And vascularized tissue can provide a way to actually restore the proper function of the heart. So those are just two potential applications for this technology.
So this all sounds pretty good, but why should the average person care about this?
Certainly. Well, vascularized tissue, as I've alluded to, is something that we all have. And unfortunately, we all know folks in our families who have had to deal with, say, cardiovascular disease or neurological diseases. And if we can better engineer models of these different diseases to study these issues potentially in a dish, then perhaps down the road we can develop new and improved therapies for, say, cardiovascular disease or neurological disease or even liver function. So these are translational applications for vascularized tissue, and I think that really the sky's the limit for this type of technology.
That's a future I want to be part of.
Certainly.
I want to know how you got into this challenge, and were you surprised to learn that NASA was even doing this challenge?
Well, I've been excited about the intersection of space biology and microgravity research and cardiovascular research for a long time. I actually had the opportunity to send an experiment to the International Space Station a few years ago, studying the impact of microgravity on the human heart using stem cell derived heart cells, which are really exciting because you can see them beat in a dish, not something that you see every single day. And just so happens, the astronaut we were working with on that project is Dr. Kate Rubins, who's not only an outstanding astronaut, but also a cell biologist. And I believe we'll be talking to her in a few moments. So we actually collaborated in the summer of 2016 to send these samples to the space station for a period of about 1 month, during which time we actually studied how the human heart and the cells of the human heart can change during long-term exposure to low gravity. And this was an exciting experiment, and we learned a lot from it, but we're always trying to figure out new ways to improve these results and improve the subsequent experiments. And one thing that was really missing in this experiment that I did, back in the day, was a vascularized network of tissues, a vascular tissue, right? So this was just looking at isolated cells in a dish, but as I've mentioned, the real human body is comprised of many, many blood vessels. And so if we want to better understand how the human body is going to function in low gravity, we have to use these next generation models that contain these blood vessels.
Wow, you have given us a lot to think about and a lot to be excited about. And I want to thank you for joining us today. Thank you so much.
Thank you again for having me.
The benefits of this technology are so exciting for medicine here on Earth. But what about in space? We are now here with Robin Gatens, director of the International Space Station. Robin, I'm so happy you could join us today.
Thank you for having me, Lauren.
All right, let's get straight into it. What technologies, or what do technologies like those being solved by the vascular tissue challenge mean for space exploration?
They're important for a couple of reasons. One of the key missions for the International Space Station is to use it as a test bed to enable future explorations beyond low Earth orbit. So the vascularized thick tissue models resulting from this challenge will function as organ analogs, or models that can be used to study deep space environmental effects such as radiation, microgravity deconditioning, and then help us develop strategies to minimize the damage to healthy cells and mitigate the negative effects on humans for these long duration missions away from low Earth orbit. One of the other key missions of the International Space Station is to create new markets in space. We're trying to create an economy in low Earth orbit where NASA is one of many customers, and there's all kinds of other enterprises going on. And so we think the area of regenerative medicine and tissue engineering is one of the most promising of these potential applications that can create sustained non-NASA demand for ongoing research in microgravity. And in microgravity, it could be possible to print or grow tissue or even organs better than we can here on Earth.
That is wild. Who would have predicted that? I guess our scientists. So we all love our astronauts on the ISS, but most of us are here on Earth. So how can developing technologies for space affect us here back on planet Earth?
We have so many technologies we've developed for space that has spun off to applications here on Earth. And that's another key goal of the International Space Station, is to do research that benefits life here on Earth. And so on Earth, the vascularized tissue could be used in pharmaceutical testing or disease modeling. And it could also accelerate new research and development in the field of organ transplants, as we talked about. Other areas that we have developed technology for, another example is in our water recycling technology that we use on the space station. That technology has spun off into ground-based filtration systems that have been used in disaster relief and remote areas where there is no clean water.
Wow, this makes me very excited and hopeful for the future. Robin, I want to thank you for joining us today. We've really loved having you.
Thank you. I'm excited to have participated. Thanks a lot.
Of course. Speaking of space exploration, let's hear from someone who knows firsthand what living and working in space is all about. Coming to us from space, astronaut Kate Rubins. Take it away, Kate.
Greetings from the International Space Station. I'm Kate Rubins, Expedition 64 flight engineer. I'm a NASA astronaut and a microbiologist. And I want to congratulate the NASA Centennial Challenges team and all of the participating competitors on the success of the vascular tissue competition. Research conducted here in microgravity helps NASA prepare for long-duration missions to the Moon and to Mars, and contributes to improvements for life on Earth. During my first spaceflight mission in 2016, we were able to sequence DNA in space for the very first time. With this breakthrough in three-dimensional printing thick, vascularized human organ tissue, we could be able to test the impacts of microgravity on tissues right here on the International Space Station. And this could pave the way for medical breakthroughs in tissue preparation and regeneration back on Earth. I'm excited about the research being done by all of the Vascular Tissue Challenge participants, and I really look forward to potentially working with your tissue samples here on the International Space Station in the future. Thank you, and congratulations.
Thanks, Kate. We are moments away from announcing the winner of NASA's Vascular Tissue Challenge. But first, let's hear from the Methuselah Foundation, an organization that partnered with NASA to help make this challenge a reality. We are joined by Dave Gobel. Thank you for joining us today, Dave.
It's my pleasure. Thank you for having me.
It's wonderful to have you. All right, so let's, lay some groundwork here. Why did you team up with NASA to support the Vascular Tissue Challenge?
Well, we're a prize funding organization as well as NASA. About 6 years ago, we had a prize for the creation of a whole liver that would be viable in a model organism for 3 months, keep it alive, keep it healthy. But we were way too early, and the technology didn't support that goal. And so we stepped back and we asked ourselves, what was the real rate limiter for getting whole organs? And the real problem was that you could not scale the tissues up. Above 20 cells thick, the tissues would begin to die because of a lack of blood flow. And that blood flow was provided by microcirculation, tiny capillaries and vessels. And nobody knew how to make those. And so we thought we would propose to NASA the creation of a prize for microvasculature. And NASA, after a couple of years, said yes, and so here we are.
Amazing. Yeah, the rest is history, right? So where do you see technologies like growing human tissues and human organs in 10, 20, 50 years from now?
Well, in the next 5 to 10 years, we will be able to make tissue patches to print out, for instance, liver tissue that would be added to a sick liver, and that liver could potentially recover itself because of the crutch-like capability of the liver patch, or it would allow for more time for a transplant to become available. So that's actually in a reasonably near-term future from a technology standpoint. In the next 10 to 15 years, I would say that we would be able to get whole livers and whole kidneys. And the reason we want to do that is because there's 110,000 people always on the waiting list, and 25% of them pass away while they're waiting. But there's another 10 times that don't qualify to even be on the list for an organ replacement. So we want to make that all go away, and we want to give the surgeons all the inventory needed to make it possible. And then shortly thereafter, the organs will be composed of your own cells, so that you'll never have to be on an autoimmune drug. It will just be a new you, at least as far as that organ is concerned.
Wow, think of the millions of people this is going to affect. This is incredible. I want to thank you for joining us today. We absolutely loved having you.
Well, thank you very much, and thanks to NASA for sponsoring the prize.
Absolutely. As we've learned, solving the challenge of engineering vascularized tissue can have far-reaching benefits for both NASA and the world. Let's explore more.
The vascular tissue challenge is pursuing the idea of creating a slab of tissue with its own blood supply.
This is going to be like a light switch. At the moment, we can make tissues, but we're not good at vascularizing them.
The benefits of creating vascularized tissue are enormous, and they're far-reaching.
It would really revolutionize and expand our ability to treat a multitude of diseases involving all the different organ systems.
And also for screening toxic effects of different drugs.
In the long term, there's even the potential to create whole organs that could be used to transplant. So why is this technology important to NASA?
It would open up the possibilities of extended space travel.
We could use them to look at the effect of things like gravity that could enable us to advance the space program.
Without sending someone into deep space or having to recreate that environment in toto.
And at the end of the day, improve lives for everyone.
That's why we need something like the NASA Centennial Challenge.
It can really change the face of medicine as we know it.
And now for the exciting moment we've all been waiting for. Here is NASA's Associate Administrator for Space Technology, Jim Reuter, to reveal the winner of NASA's Vascular Tissue Challenge.
On behalf of NASA's Space Technology Mission Directorate, the Prizes, Challenges, and Crowdsourcing Program, and the entire agency, I'd like to congratulate all of the participants of the Vascular Tissue Challenge. You know, when we set out to open this challenge in 2016, we couldn't imagine the success we'd see from these teams. In fact, I recall us talking about that this challenge to produce thick, metabolically functional human vascularized tissue was maybe a step too far and could not be done in the time allotted. But these teams have worked through many setbacks associated with a global pandemic, often with limited or no access to their labs. Their dedication to the medical and space communities is an example of how, even through adversity, the American people will persevere. With the ability to regenerate thick, vascularized tissue, we can make an enormous impact on Earth and in space. And so it is my pleasure to announce the first place winner of the competition, Team Winston from Wake Forest Institute for Regenerative Medicine. Team Winston will receive $300,000 from NASA for their innovation. Additionally, thanks to the ISS National Lab, managed by CASES, Team Winston will have the opportunity to test their tissue under the effects of microgravity on board the International Space Station. Selections for competitions like these couldn't happen without our judges who are truly experts in their field from across the country. I'd like to thank each one of you for lending your expertise and for ensuring our participating teams successfully created tissues that we can use to study deep space environmental effects. So again, congratulations to Team Winston on your achievement, and thank you to everyone involved in the competition for your contributions to advancing space exploration.
And we are here live with Kelsey Wilson from Team Winston, the first place winners of NASA's Vascular Tissue Challenge. Congratulations, Kelsey and team.
Thank you so much for having me on here.
It's really exciting to have you after all this challenge had to offer us. And joining us with Team Winston is Vascular Tissue Challenge judge, Dr. Arun Sharma. Welcome back to the show, Dr. Sharma.
Thank you for having me.
Okay, so we've got lots of questions for our two guests. The first one is, you're the first person to accomplish this task, Kelsey, you and your team. How does that feel? How are you and your team feeling right now?
Well, for me personally, it feels a little surreal. We work on these projects for years and you spend hours and hours in lab, but it is absolutely amazing to get something all the way to the point that we can really bring it to the public. So that's been very exciting. And to be honest, when I started grad school 4 years ago, I certainly never imagined that I would be being interviewed by NASA. So yeah, I think surreal.
Absolutely. Dr. Sharma, I understand this is a pretty remarkable accomplishment. having judged this competition, how much of an achievement is it to finally have solved this incredible feat of engineering functional human tissue?
It's a tremendous accomplishment. It's not easy to create a portion of tissue that contains blood vessels, is able to survive for a month, and is still able to be functional. Just meeting one of those criteria would be an accomplishment, but the fact that Kelsey and her team were able to meet all of three criteria is absolutely tremendous.
Kelsey, tell us about the winning tissue sample and the approach that you and your team took to solving this problem.
Of course. Well, we started out with some of the criteria that was laid out by NASA and the Methuselah Foundation, really starting with it needed to be a centimeter in each direction to make it really large and something that we could potentially use, for studying whole organs. And then we took a pretty stepwise approach. We looked at what shape would be the best for getting fluid flow all the way through the samples and for getting nutrients in and out of it, and settled on a gyroid shape, which is a series of interconnecting wave-like tubes. And then we looked at the best gel for allowing our cells to really grow and expand and communicate with each other, which ended up being a modified gelatin, sort of similar to what you might make Jell-O out of at home in your kitchen. And then building a circulation system that we could get fluid flow through and, really allow these samples to be perfused in a way similar to what you see in the human body.
That is a use of Jell-O that I never thought would be applied in this context. Dr. Sharma, you've been a judge for this challenge for nearly 5 years now. In addition to the work that you do, what do you get out of judging this competition, both professionally but also personally?
Right. So personally, it's very gratifying to see the incredibly talented young minds around the country coming together to address this very unique problem. So it's tremendous from the perspective of a judge to see all of the unique solutions that the different teams have come up with to actually address some of these concerns when it comes to tissue functionality and tissue vascularization. But if we're talking about the long-term medical applications, something like this is extremely important. Down the road, perhaps some of these vascularized tissues may be able to serve as replacement tissues for tissues that are lost due to injury or disease. So this is a critically important challenge. And again, I wanted to congratulate Kelsey and her team, as well as all the other teams, for their tremendous work.
Indeed. Kelsey, what was it like to work with NASA? And would you recommend that experience to others?
Absolutely. I think NASA was incredible through this. Not only was this a scientifically difficult challenge to work around, but also, with COVID in particular, we had quite a few technical difficulties. The judging committee, for example, couldn't come out and follow us around labs, so we had to do that all over Zoom, and they were absolutely wonderful, helping us both along with the scientific challenges that we came up against, but then also certain technical hurdles that we had to overcome.
Yeah, COVID alone would have been a serious hurdle, but everything else, on top of that makes it an incredible feat. What's next for your team? Do you get a break now? Is this sort of, the only thing you were working on? What's up with your lab after this?
Well, for those of you who aren't in research, it's pretty rare for any one of us not to have a couple different projects going. So I know that the team A is continuing this work and gets to send it up into space, which is very exciting. But one of the other major members of this, Dr. Moon, is working on taking this gyroid and applying it to neural tissue to look at possibly developing a blood-brain barrier prototype. And I myself am going to be able to spend a little bit more time focusing on some of my work developing 3D printed skin and adding hair follicles into it. So a lot of different projects.
It sounds like it. That is incredible. This question, I think, would be interesting coming from both of you. Not every team took the same approach, obviously. There were many ways to solve this challenge. Could you talk a little bit about that, what you did, Kelsey? Because you were on the winning team and the second place team, and our judge here probably saw many approaches as well. So Kelsey, if you could take that first.
Well, of course. As you just said, we actually had 2 different teams working on this at Wake, and we both took very different approaches to it. Even though we were using bioprinting, we used 2 different types of bioprinting, and came up with structures that looked very different, despite the fact that they both met the challenge requirements. So, even within the Institute, we had multiple ways of attempting this, and I can't wait to see what the other groups came up with, because I assume that they are outside of my imagination and probably just wonderful.
Absolutely, you're totally right, Kelsey. And as a judge, I've had an inside look into what teams around the country have been able to do, and it's astounding, really. All of these exceptionally talented bioengineers have utilized a number of different approaches, including 3D printing, for example, to create custom tissues that have a variety of different cell types. We had teams utilizing liver tissue, heart tissue, and so on. And the possibilities are really endless as to how the cell types can be properly integrated and intersected with the tissue engineering approaches.
And actually, I'd like to piggyback off of that. What did it take for for a team to win this challenge, Dr. Sharma? What basically was unique, different about Kelsey's team?
Right, so Kelsey's team was the first team to successfully meet multiple criteria that are associated with the success of this challenge. As I alluded to, this was a very difficult challenge in the first place, but the fact that you had to meet multiple criteria made it even more challenging. So one criteria was functionality. The tissues that were actually developed have to be functional for an extended period of time, in 1 month in particular. Also, these tissues had to be vascularized, so they had to contain the cells that line the blood vessels in the body. And they have to survive. They have to appropriately survive long term. Again, our timeline was at least 1 month. And Kelsey's team was the first to accomplish these tasks and to meet these criteria. So an exceptionally challenging task, but congratulations again to your success.
Yeah, I'm curious, we've been talking about how challenging something like this is. And this is a question I'd like to ask of you both. What motivates you to do this kind of work? Kelsey, you and your team, and then Dr. Sharma, you judging, but also the work you do outside of judging. What motivates you to do this? Kelsey, let's hear from you first.
Well, I think that there's, a lot of different motivating factors. I know for me personally, some of this is getting to be able to have something that will really help the research community, be able to study diseases and different disease states and even healthy systems, and maybe be able to come up with something that we can use to test different medicines on rather than having to go to animal or straight to human testing. But also it gives a really unique perspective into a building block that, as has been talked about earlier in this segment, I'm really hopeful can eventually come up with whole organs that can be used for organ replacement in the future. So building blocks.
I agree with you, Kelsey. Absolutely. Building blocks for the next step. I think ultimately the hope in this field in regenerative medicine and bioengineering is to design pieces of tissues that may be able to one day replace tissues that are damaged or injured. I think it's a dream, and it's something that can positively benefit the lives of millions of people around the world.
That's incredibly exciting. And what else is really exciting is that I understand, Kelsey, your sample will fly to the ISS for further testing. So what are you hoping to gain from testing at the International Space Station?
Well, I think the first thing we're really hoping to look at is how space affects this. I mean, we know that humans react differently in space conditions than they do here on Earth. And there's been a lot of tests, as Dr. Sharma has talked about, looking at cells being grown in microgravity and under, other space conditions, including radioactivity. But those have confounding factors. They aren't fed via flow. They're in a static media culture, and that microgravity system has been shown to change how much oxygen they're able to get into that culture. And it will be, personally very interesting to look at how the space environment actually ends up affecting a system that's fed in a physiological manner.
And Dr. Sharma, as
Absolutely.
someone who has done research in the past on this, I would love to get your take on that as well.
Certainly. So as I alluded to previously, the work that I've done is in two-dimensional cell culture, so cells grown in 2D, and really the human body functions in 3 dimensions, right? So that's part of the reason why this challenge is so exciting. We want to be able to create better models of how the human body is actually going to function in low gravity during extended space flight. So a 3-dimensional tissue will be able to better tell us about exactly that, the impacts of low gravity on the human body. In addition, the International Space Station as a research laboratory is largely for the benefit for those of us here on the Earth. So we want to model some of these phenomena, such as accelerated aging, because in a lot of senses, in a significant way, the phenomena that you see in low gravity and during spaceflight is a form of accelerated aging. The bones of the body are deteriorating at a slightly faster rate, the muscles as well. And so what we learn from these studies of in vitro models, of 3-dimensional, 3D printed models, will help us better understand aging, something that we're all going through here on Earth.
Yes, 100%. I'm not the only one asking questions here. All of you have been asking questions online since the beginning of this episode, so let's get to those. Don't forget, you can ask your questions by using the hashtag AskNASA or by writing in the comment box wherever you are watching this. Okay, so our first question is from Space Girl 123, that's cute, on Twitter, and she asks, why is NASA interested in engineering human tissue? Arun, why don't we throw that one to you?
Surely, absolutely. And just like I alluded to, NASA is really interested in this because we want to understand ways that this tissue can perhaps improve the functionality of tissues on the planet. So we perhaps this tissue will be better, more functional in a microgravity environment. That'll tell us more about how we might be able to utilize this tissue for those downstream applications of cell therapy, cell replacement, tissue replacement. I think there's a lot we don't understand about the microgravity environment and its impact on the human body. And importantly, we want to figure out the beneficial effects that microgravity might be able to have on human tissue function.
That makes sense. Moho Gamer on YouTube asks, if we can find a way to make these types of tissues, will it be able to create organs? So I think Kelsey talked a little bit about this, but I think the idea here is, can we create an entire organ? Kelsey, do you want to take that one?
Sure. Well, eventually. I won't say that we're going to create a liver in lab next month. That might be a little far out for us. There are several things that need to be optimized before then, including, for example, how do we get enough cells to be able to print something that large? Growing cells is a hurdle that also needs to be overcome. Next potential NASA challenge. But it certainly is a first step, and one of the next goals is taking this and possibly moving on to something bigger than a centimeter.
Arun, did you have anything to add to that?
Yeah, I'll echo what Kelsey just said. I mean, a lot of what we've done here is incremental, but extremely important. The next step is to make these tissues even bigger and better, larger than 1 cubic centimeter, as Kelsey had mentioned. If the organs of the body are, of course, larger than that for the most part, I think it'd be a dream to perfectly replicate a truly adult human organ outside of the body. We're not quite there yet, but down the road, who knows, we might be.
So Sammy on Facebook asks, why did you choose liver cells? And I'll throw that one to Kelsey. And actually, I'm very interested in the answer to that as well. Why liver cells?
Well, there was a lot of talk between the different samples that we could look at. NASA and the Methuselah Foundation had outlined 5 of the major different organ types that they would like groups to look at, including liver, kidney, skeletal muscle, heart muscle, and lung. And I think one of the reasons we wanted to look at liver is it is a large solid organ, which means that you can sort of develop it with relatively few really tiny components that you have to get in there. I mean, with lung, for example, you have to build in an air-liquid interface. With cardiac muscle, you need to make sure that you can electrically stimulate it. And so liver kind of acts as a first step towards being able to move to some of these that might have more cellularly complex systems, while still being something that's critically important. I mean, liver failure in our country is devastating to many, many people. And so it's an important step, and also sets us up for being able to succeed with more complicated structures.
Our bodies are just amazing. It's incredible. I think this would be a good question for Dr. Sharma. Hayden Ravenscroft on YouTube asks, are there any experiments running on the ISS or in space regarding vascularized tissue right now?
So right now, there are not, as far as I know, there aren't any vascularized tissue experiments running on station currently, but that may change very soon thanks to the tremendous work of Kelsey and her colleagues. Most of the work that has been done on station has been either in animal models or in two-dimensional cell culture systems, and some organ chip related systems as well. Three-dimensional vascularized tissue, for one, is very difficult to create. So this particular challenge was addressing that. Can we actually make something like this in the first place? And the next step of this will be, yes, transporting it to the station and really utilizing it as an appropriate model to study the function of the human body.
So we talked a little bit about this before, Kelsey, but how did COVID impact your team's participation in the challenge? Did it extend the timeline? I know you said before that the judges couldn't come out and actually look at the sample. How else did your team sort of overcome this unprecedented challenge?
Well, COVID was quite a hurdle. I mean, they actually shut down the lab for several months and wouldn't let any of us show up, although I know a few of us may have snuck in when we weren't supposed to be there. It's very hard to let go of research that you've spent years working on because of outside problems. I think it gave us a little bit more time to plan what our final judged experiment would be like. I know that what you're seeing here is one experiment that worked really well, but this was years of progressive failure leading up to it, and learning from what didn't work well to move forward. And so it gave us some time to sort of synthesize all of that together and come up with really how we wanted to try to put this all together into a real working project.
Yeah, I think you raise a really excellent point. And we spoke a little bit about this with Dr. Sharma, where, this is like 5 years. This is at least, his participation. And this has been an ongoing sort of, like failure after failure that eventually results in something that works. And I think that's a really important message, that like sometimes something worth achieving takes a very long time, but it is worth it in the end. What was something that really surprised you about your results, Kelsey? Did everything go as expected, or were you kind of surprised by anything about the process?
I think working with living tissue, nothing ever quite goes as you expect it to. It always does something a little bit different than you thought it was going to do. I think one of the things that we were really, really excited to see with these samples was the fact that they aggregated so well, which means that the cells came in, instead of being individual cells, they really merged together into something moving towards an organ, where it wasn't just cells in a gel, it was really cells coming together and starting to make a precursor to tissue. And while we were expecting some aggregation, I think we saw a bit more than we were hoping for. So it was a really great thing to see.
Great.
Yeah.
That's a great problem. I think a lot of people are wondering, for both of you, how you got into this line of work. I think sometimes you sort of fall into it, sometimes it's very intentional, sometimes it's both. Starting with you, Dr. Sharma, how did this line of work begin for you?
Right. Well, I've always had a passion for spaceflight and the intersection of biology and space. I actually had the good fortune of growing up in Huntsville, Alabama, which is just down the road from the Marshall Space Flight Center. And I think part of it is a pursuit of the unknown. There's a lot we just don't know about how the human body is actually impacted by microgravity. I mean, we've been in space for a few decades now, and we have an understanding of the physiological changes that happen at the body level. But as a cell biologist, I'm really interested in the cells. How are the cells of the human body? How are the genetics of the body impacted by extended space flight? And I think, right now we're kind of at a golden age when it comes to utilizing the space station to better understand some of these questions about the impact of microgravity on the human body.
And what about you, Kelsey?
Well, for me, I mean, I've always loved math and science and how if something doesn't make sense there, you just need to dig a little bit deeper to find a foundational theorem that sort of ties it all together. Being able to take that and move it into biomedical engineering was a way to bring that idea into something that actually helped people in a really primary way, rather than, I mean, so many different technologies these days help people. It would be crass of me to say that other groups are not, but with biomedical engineering, I can really see the people that I'm helping directly. With regards to this particular project, one of the reasons I came back to graduate school was that I really thought bioprinting was one of the ways that we could scale up this system and actually get it to patients. I'd spent some time doing research where we were making different implants by hand, and that just was never going to actually get to people, in my opinion, not in a large scale way. And for this particular project, I can't speak to how each of the other team members came on board, but for me it was when I showed up at school and said I wanted to work on bioprinting, Dr. Atala had asked what system I wanted to work on, and I said, "Do you have an unsolvable problem? I like those." He gave me this.
Oh my goodness. Wow, that is fantastic. I think we're all trying to do our part here, and we're all trying to do it in the best way we know how. Here's a question that I think a lot of people are thinking. Mark Hockmeister, sorry if I pronounced your name wrong, on Facebook says, could this extend our lifespan? I think either person can maybe speak to this. We'll start with Dr. Sharma first.
Sure. Well, I think that is the dream, as we have talked about a lot here on this segment. I mean, down the road, if we can perhaps engineer replacement human organs outside the body, then certainly that could extend lifespan. Say if you have a failing liver or a failing heart, you might just be able to replace it entirely by growing a personalized organ outside of the body. So this challenge is a really important step towards making that dream a reality.
I completely agree with Dr. Sharma. I don't think this particular construct is going to save a life the way that it is, but I'm really excited that it's, a step towards moving to full organs or potentially developing new medicines and new treatment plans that could help with saving people.
So I think what we're saying to Mark is, let's get this sucker on the ISS and then we'll start there.
Let's do it.
Yeah. Okay, so I'm curious, what are the current limitations on creating new tissue? And I think you probably both could speak to this. Like you said before, this was a process that took years in the making. What kind of I can't imagine where one begins to do this sort of task, but what kinds of limitations exist when you're trying to start?
Well, I can take a stab at that question. There's quite a few. I think the very first one you have to start with is creating an environment that the cells are really going to be happy in. And there are so many different factors that lead into that. Different cells like different stiffnesses, different cells like different amounts of different chemicals to help stimulate them and make them mature into certain ways. And so, I mean, for example, in this tissue, we used two different cell types, endothelial cells, which make up your blood vessels, and hepatocytes, which are the primary cell found in your liver. And endothelial cells need very different growth factors than hepatocytes. So we had to start blending different medias together to create a substrate that would feed both of them and keep them both functional, developing a system that creates flow. And then, as I alluded to earlier, your liver, or really any one of your organs, consists of billions and billions of cells. So being able to grow billions of cells in the lab is really difficult. So being able to scale up and create that number of cells without months of growth protocols, or a scientist like me going in and feeding them every couple of days with tons and tons of media, is an important thing that we need to overcome.
Absolutely, and I think
Kelsey.
yeah,
Go ahead, Dr. Sharma.
I think Kelsey is totally right here. And in addition to those issues of functionality, survival, one thing that we're still grappling with now, and something that wasn't really addressed at all in this particular challenge, was appropriately integrating these tissues into the body. So if we were to make, say, a liver outside of the body, how do we ensure that once we put it back into the body, it's going to function appropriately, integrate into the host's own vascular network, and integrate with the host's immune system as well? These are incredibly challenging issues, but I think Kelsey and your team, you're going to be busy for a while.
Yeah, sounds like it. Lots of work that's still ahead of you, but all good stuff. We're coming to a close here, but I want to ask a couple more questions. Dr. Sharma, what was the most important thing to you about participating in this challenge? We talked to you a little bit about what it personally meant, but this is something that we said before. You've dedicated now nearly 5 years to this. I mean, why?
Yeah, it's a fantastic question. I think again, it comes back to pushing the boundaries of science. I think part of the reason you're interested in science, you get into it in the first place, is perhaps you want to make a benefit for human health and you want to improve lives down the road. And to me, I mean, I was a judge in this challenge. I wasn't actually participating, unlike Kelsey and her team. But for me as a judge, it was really rewarding to see the work that Kelsey and her team and other teams around the country were doing to help perhaps address this unmet medical need of replacement human organs. It's a vision and a goal that we all want to strive for in this field, and that's why something like this is so critically important, because challenges like these and experiments and projects like these are going to make that vision a reality down the road. So it was very rewarding to be a part of this.
Ooh, I felt that one. That was a great answer. Okay, so we've got a very practical person here asking this question, and we'll just do quick answers from you both. What advice would you give to future solvers interested in a NASA challenge? So from both the participant side and also the judges side. Quick answers. Let's start with Arun.
Well, I would say a passion for the field, a passion for what you're doing. You don't necessarily have to be directly involved in space science. I think being passionate about engineering, passionate about biology is critically important for success in this challenge or in future challenges like it.
I completely agree with Dr. Sharma. Passion is definitely one of the most important things. Learning how to give up a little bit of sleep for a while is probably also pretty critical. Make sure you can get all the assays done and, get everything turned in on time.
Hey, that's I appreciate your realism there. Well, on that note, that is all the time we have for today. Thank you so much to you, Dr. Sharma, for being here.
Thank you so much for having me.
And congratulations again, Kelsey, and to your entire team. Congratulations.
Thank you so much. I'm honored to be here as a representative, and it's certainly been a major team effort. Very exciting to be honored in such a way.
Absolutely. So today we learned all about vascularized human tissue, the very existence of which we need to survive. We met the very first scientists to create thick, functional, vascularized human tissue in a lab. We have explored the many benefits of this technology, both on Earth and in space. Now let's imagine the possibilities for what's next. We're also excited to announce that the judges have confirmed a second place winner as well. Team W Firm, also from the Wake Forest Institute of Regenerative Medicine, has taken the second place prize, winning $100,000. Not too shabby. So congratulations to them. You can learn more about Team Winston and Team W Firm technologies, as well as other competitors in the challenge during a live virtual summit coming in August. Follow at NASA Prize on Twitter and Instagram for details coming soon. And the Vascular Tissue Challenge is just one of many challenges going on at NASA right now. Crowdsourcing ideas through competitions is one of the many ways NASA advances technologies and solves difficult problems. Some of the challenges going on right now are the Deep Space Food Challenge, NASA Tech Rise, the Break the Ice Challenge, and so much more. See all of these opportunities for how you can get involved at nasa.gov/solve. For NASA Science Live, I'm your host, Lauren Ward.